Go to the white matter because tau is obviously in axons.
So tau is a normal protein that is produced in our brain at very high levels, and its function appears to be in stabilizing nerve cell processes or their connections. It's a very important molecule in normal brain function. But in certain diseases, it tends to clump together. The parts of the brain where that occurs begin to shrink, and nerve cells die. And so when that happens, if it's in an area of the brain that controls memory, then you lose your memory. If it's an area of the brain where that happens, where it controls speaking, then your speaking isn't normal anymore. Tau in the blood is very low. It's very difficult to measure, but you can measure it at a very, very low level. And what we found in this paper is the reason it is so low is once tau gets into the bloodstream from the brain, it is rapidly degraded by the body. When you give an antibody to tau, to either a mouse model that develops tau accumulation, or to a human being, that tau in the blood increases a lot over its baseline levels. So it goes up around 100 to 200 fold higher in the blood after you give the antibody. And this occurs rapidly over hours. We also found that the source of the tau that's increasing in the blood is coming from the central nervous system or the brain. Perhaps by measuring it in the blood, that might provide some insight into what's going on with tau in the central nervous system or in the brain. If we end up finding the same thing going forward in human beings, then it might be that by measuring tau in the blood after giving an antibody or something like an antibody, that might serve as a test to determine whether somebody has a particular disease or not, like Alzheimer's. And right now, there's not an effective blood test for Alzheimer's. The tests that are available to assist in diagnosis are relatively more invasive, either requiring spinal fluid analysis or imaging using radioactive tracers, where you have to do a relatively expensive test called a PET scan to determine whether somebody has the molecular features of the disease.